All right. So there's two topics I'm going to really fly through. So spatial stochastic with SmolDyn. This is in virtual cell. This is Steve Andrews, the creator and maintainer of SmolDyn. We integrated SmolDyn like 10 years ago into virtual cell and the integration was non-trivial to merge those two frameworks. Um, it's based on Smolakowski theory, which is a way of describing chemical kinetics in a macroscopically observable way and having it apply to particle Brownian dynamics. And maybe I'll skip over the theory, but I find it interesting because the derivation of the Smolakowski parameters is derived using reaction diffusion equation. So it really it has a certain amount of intuition. Um, but I'll skip over that. Uh, what's the motivation? When is spatial important? When is stochastic important? Stefan just discussed when stochastic's important. When spatial's important really is when you have some localization of sources and sinks and different structures, right? If everything's uniform, you don't need that. Spatial stochastic is when the influence of a few molecules has has a large impact, maybe with channels and pumps or other things like that. And then hybrid spatial stochastic, which we also provide, is when we have some parts of the model, say, are channels or pumps that are few and have large impact, and other parts of the model, say, like electrolytes or other regulation, is well described in a continuum. So we have the ability of doing PDEs coupled with particle Brownian dynamics. Here's a just a fly through of two um, two models, and on the right you see the SmolDyn representation. The left you see the traditional PDE from virtual cell. Here's a uh, kind of a toy model of channels that are opening and closing and diffusing within the membrane. And when they open, there's a large impact on local concentration. So we're actually looking at, I think, probably calcium or some ion near there. And this was based on work that we did with Boris Savchenko and Igor Novak and Fei Gao and myself many years ago. Um, and here you have an example of a cardiac myocyte where you have sarcomeres and T-tubules and local activation. And especially in the case of sparks, you can get in the continuum approximation, sparks will never happen. Now, you could also say that sparks never happen in reality anyway, but that's a biology topic. But occasionally a random opening of channels can have large events. It can propagate to neighbors, and sometimes the whole cell can fire when it's not driven, when it's just quiescent. And this is something that also is handled nicely by the coupling of discrete and continuous in the spatial. Um, so I guess that's this is just a screenshot of some things. Well, let's see.
Yeah, I don't, let's not get into kinematics. I mean, that spatial stochastics is really a
That's the most
nice,
important thing. Yeah.
yeah, nice introduction to Margaret Johnson's talk. So you can
Okay.
spend one more minute
All right.
in this spatial particles based, yeah.
Okay. So this is essentially when you have single molecules can have considerable effect. This is a and especially in the at the scale of, say, a whole cell model. When you're talking about single molecules having a large effect at where you have um, uh, in a very small and fast time scale, then spring salad may be more appropriate, right? And spring salad, apart from using particle Brownian dynamics with sort of spherical molecules, there's springs and different sites that have interaction. Um, but I guess I don't know if there's any questions. There's
I
two
just wanted to make sure everybody realized that that hybrid, the stochastic spatial modeling you could do standalone in SmolDyn, but in fact the hybrid spatial stochastic is really unique to virtual cell, and it's very convenient when some of the species are present at high copy number and others are present at low copy number. So you can really take advantage of doing relatively faster simulations than you could do in a purely spatial stochastic modeling system.